So I'm Daniela Rhodes. I am a professor here at NTU, and I have a joint appointment between the School of Biological Sciences and LKC Medicine. So it's a great honor and pleasure for me to introduce Tim Hunt, and particularly since I knew him from my time in Cambridge. I was in Cambridge for 40 years before coming here, and also from the time he was the chair of European Molecular Biology Organization, the Council of EMBO. I sat on the committee.
Yeah, well, you got the council. I thought you were in charge of one of those.
The fellowship program.
Most important.
Yeah. So Tim was born and grew up in Oxford, but then defected to Cambridge to do a degree in the natural sciences. He also did his PhD in Cambridge and then stayed there for almost 30 years. For the first 20 years or so, he was actually interested in understanding how proteins, or translation, how proteins are made from RNA. So in this sense, he has actually a common research interest with the speaker tomorrow, Ada Jonat, who worked on the who will tell us about the ribosome. But so Tim's interest switched in research instance changed when he in 1982 or thereabout, he discovered cycling whilst teaching in a summer course at Woods Hole. So my memory actually of Tim is of this long-haired guy. You did have long hair, right? Tall guy, spending a lot of time in our canteen at the MRC Laboratory of Molecular Biology, whereas he was actually had a position in the biochemistry department. You didn't? 19. Okay. I did try and find a picture of him with long hair, but I didn't. And I also remember actually a conversation, a coffee time conversation with Aaron Klug, and it must have been in about 1990 when Aaron was telling us that Tim Hunt was leaving Cambridge to go and work at what at the time was called Imperial Cancer Research Fund's laboratory in Clare Hall outside London. And he said something which at the time, I couldn't judge. He said, the biochemistry let the best man go. And of course, Aaron turns out to be right. And Tim went on, as you know, to win the Nobel Prize for Physiology or Medicine in 2001. And he shared the prize with Lee Hartwell and Paul Nurse for their discoveries of the key regulator of the cell cycle. So let us welcome Tim.
Thanks, Daniela. So I feel a little bit like a fish out of water here, actually, because what do I know about emerging patterns? Not a lot. So. And I don't even, one of the first rules of giving talks is you should know who you're talking to. And I don't really. You're a fairly unknown audience. And from what I've heard this morning, oh, that would be great, yes, because I like to walk around. Sorry. This goes in the pocket. That goes in my pocket? Goes in my back pocket. Okay? Is that on? Oh, you've got to switch it on. Oh, where's it? In my pocket. Oh, you told me to put it into me. It's all right. Oh, brilliant. Thank you very much. That's fabulous. Now I've lost completely what I was thinking about because of that. Why am I here? The audience. Yes, no, I don't know the audience. And judging by the talks this morning, these were very interesting, but boy, what a different landscape. What a different, what are those musical people? soundscapes, I think they're called. Sound worlds. Sound worlds, that's what they talk about. Well, it's very different sound worlds. I'm afraid that this is going to be very down to earth. And I thought what I would do is, because of, I think, the heterogeneity that I think I rightly detected ahead of time, I'm going to start off with a rather simple biology lesson. So apologies to the biologists among you, but it's as well to remember this. And it came. I just was in Tokyo before I was here on Sunday. The Nobel people, they're getting very funny ideas. Anyway, this was what would they call them? Nobel dialogues. So we had loads of round table discussions in audiences, and from some of the questions that the audience asked, and this was about genetics, I realized that there are some fairly fundamental misconceptions among the general public about how life works. So, let's start. So first of all, we have this biology lesson. Then a little history of discovery, how you could call it sort of how to win a Nobel Prize. And then at the end, some stuff about really what I see as the problems of complexity in biology. And it'll look very, very simple, but it turns out to be very, very hard to understand. And that's based on the work that I did with a wonderful Japanese collaborator right at the end of my career. I'm retired now. I don't have a lab anymore, but I still take an interest in this stuff. So I'm going to sort of try and, it's a bit of a shaggy dog story. And I think I should sort of preface my remarks by telling you the story of the ambassador. I quite often give a talk, and the ambassador is there. Now, ambassadors, as you know, are very diplomatic. They're also very intelligent, but they are never scientists. So when I've given my talk, the ambassador always comes up and says, "Tim, that was an absolutely marvelous talk, but I didn't understand any of it." And I've never been able to get the ambassador to explain exactly what it was that they're always he's, in my experience, didn't understand. And I'll have just one or two comments about that to make at the end. I've got some wonderful quotes from a wonderful, alas no longer with us, French physicist called Pierre-Gilles de Gennes, where he addresses some of these issues of the problems of communication, because I think they're quite relevant too and fun. So this talk is called Switches and Latches in the Control of Cell Division. And the first thing we really need to talk about is cells, because that's fundamental. So remember, everything is made of cells. And cells are very, very small. And cells only ever come from other cells. This is a picture I took very recently in Berlin. It's Rudolf Virchow in his museum, a small fraction of which survived the war. And I should surely show you some lovely pictures of what's in that museum, but some of them are pretty disgusting. So what Virchow pointed out at the end of the 19th century is that cells only ever come from other cells. And that has, that, if you like, is a sort of the atomic theory of biology. It means that if you believe that, it's very hard to understand where the intelligent designer might have come into the picture. Did the intelligent designer affect the neutrino flux in the first 10 to the minus 18th seconds of the Big Bang? Or was he some kind of genetic engineer who arrived a little later on the scene? They criticize our theories, but they never produce one of their own. And if you really understand that cells only ever come from other cells, you really don't understand the first thing about biology. Now, here's another important thing, and this came up at dinner last night. And it's a quotation from this man here, who I'll show you a picture of sort of his great contribution. He's called Dan Mazia, And he was a professor at Berkeley, and he worked a lot at the Hopkins Marine Station on Monterey Bay. And he was like a sort of old-style evangelist preacher, actually. I've actually met him once or twice. More often than not, questions beginning with why are inane and of no service in scientific discourse. But in biology, sometimes they make sense. If we ask why cells must divide, the answer can be given in terms of what happens if they don't. The answer is that they die, no matter what criterion of death we apply. So not only do cells arise by division, but they have to divide. They have to divide in the same sense that probably the older among you almost certainly somewhere have a drawer full of old floppy disks with no known device that will interpret them. Yet you probably also have the files that are on those floppy disks on your present hard disk because you couldn't bear to delete them. Well, so, you know, that's exactly the same thing. The information is there, but it has to be transformed to a new physical format, and that's exactly why cells have to divide. And this is a very familiar sort of picture. This is Haeckel. What are these? These are coelenterates. And I guess Darwin was the first person to actually draw a tree and give an idea of descent, which he based, by the way, apparently on comparative linguistics. They'd been drawing trees like this for 50 years before Darwin came along. It is said, or I have read people who claim that actually that's where the idea comes. And again, the idea is exactly the same, namely that there is something which is inherited, namely your parents' language, but it's inherited with errors. So things change over time, and then they get isolated and differentiate. And it's exactly the same with organisms. And the basis of this is this magic molecule called DNA. This is a teeny-weeny structure. This, by the way, is Rosalind Franklin's model of DNA, not Francis Crick and Jim Watson's model of DNA. The information, I mean, people got very excited when the human genome was sequenced from end to end. Actually, it hasn't been completely sequenced from end to end, but never mind that. Most of the stuff that hasn't been sequenced is thought not to have very much information in it. But if you take this molecule and you sequence it, in principle, you have all the instructions for making an oak tree, a human being, a worm, whatever it may be. And it is this that is inherited, and it is this that gives the organism everything. It specifies the organism almost completely. Now, just to give you a few numbers, because I think this is quite important. So I said cells were very, very small. How small is small? Well, the answer is that you have an astronomical number of cells inside you, around about 5 times 10 to the 13th. That is a very large number indeed. And each one of those cells contains 2 meters of DNA in around about 50 molecules. Each chromosome is a separate molecule of DNA. That's a relatively new fact. And you can calculate by doing some simple arithmetic, if you were to stretch all the DNA in a human being out, how many times it would go to the sun and back. I mean, it's again, an astronomical number. And the other thing is that actually, in order to stay alive, each one of you, every second, is making about 2 million new cells, all of them produced by the division of pre-existing cells. That's mostly because of the replacement of the blood cells that die every second. But it's a prodigious number. So, and as we know from the ghastly survivors of the atom bomb explosions in Hiroshima and Nagasaki, if you're unfortunate enough to stand in the way of an atom bomb when it explodes, not close enough to be instantly incinerated, but just close enough to get a lethal dose of ionizing radiation, all cell division in your body stops, and you die after about a week in a very unpleasant way. You get anemic, your hair drops out, and most important of all, your gut fails to renew itself. But basically, you stop renewing anything. So I can't remember now exactly how much renewal goes on, but it's sort of the order of, if you take the whole body, I mean, this has been measured, there's nothing left from you from about 7 years ago. Everything is new, all turning over, all being replaced all the time. Not always by division, sometimes just by replacement of the molecules. Like, I think nerve cells don't divide, and therefore they have to be renewed in a different kind of way or supported in a different kind of way. I mean, that's a biological problem. So how does this work? Well, it's very simple, and Francis Crick said Jim had it first. So this is a Watsonian statement. DNA makes RNA, makes protein, and it's really the proteins that do stuff in our bodies. They're the enzymes, they're the stuff. If I look at your face, what do I see? Proteins. And some of the proteins are keratin, which makes your skin. Some of the rosy flush of your cheeks is the hemoglobin in your blood. The lens of your eye and the cornea, that's another protein. And, you know, as we all know, there are enzymes in your stomach that digest your lunch. But proteins can do magical things, and they're all specified in the DNA. And not only that, but there's a reflexiveness about this which gives rise to the complexity, because the proteins themselves can actually bind to the DNA molecules and then specify how much of a particular protein is made when and where. And to give you a sense of what that's like, I'd like to share with you this picture from the Wellcome Museum of Medical Monstrosities. And it's on the Euston Road, and you can go and look at it. This is a printout of the human genome. The human genome is 3 × 10⁹ nucleotides long. And I'll show you what that looks like in just a second. And this printout, I think, is in 4-point courier. And each one of these volumes has about 500 pages in it. They're divided into different chromosomes, 1, 2, 3, 4, 5, 6, down to 23 somewhere in the bottom. It's quite a big bookcase, you know, it's sort of, you can get. And if we focus on the page that is open there, you'll see what the human genome actually looks like. Now, those of you with a sort of computing bent could actually type about 18 nucleotides off that. I mean, you know, ATAA, GAG, TG, TCT, CCAT, and you would know that this was the beta chain of human globin. But looking at it, you can see it doesn't exactly resemble the works of Shakespeare. In fact, it so little resembles the works of Shakespeare that you'll notice that there's no punctuation whatsoever. There are no parentheses, there are no paragraphs, and yet, if that sequence was in a red blood cell, you would make huge numbers of copies of it and huge numbers of copies of the protein that it specifies. So somehow, this exceedingly boring-looking stuff is beautifully interpreted in the nucleus of every one of the cells in your body. And the idea is, the very simple idea, is that every single cell in your body has exactly the same DNA sequence because of the mechanisms I'm going to tell you about. But like a dictionary or a thesaurus or an encyclopedia, whatever cell you're in, you can choose a smorgasbord of genes to express, which will give that cell identity. So, you know, in my lifetime, we have really come to understand this pretty well, at least in outline. You know, how it works in actual detail is still pretty obscure. And as Watson and Crick correctly pointed out, their famous phrase, "It has not escaped our attention, ... Because the 2 strands of DNA, which I've shown you the sequence of one of them, they actually run in opposite directions. This causes students no end of trouble, and me for that matter. These two, that's not clear. You can't see the polarity of these strands. It's not marked on here. But one of them is running up, the other one is running down, and then you can make 2 daughter strands, and when a cell divides, to assure that each daughter cell has exactly the same information. So whatever cell it becomes, it can always be whatever cell it wants to be. And that's the sort of principle of the excitement in these induced stem cells. I mean, you can actually take a cell out of your skin and turn it back into an egg by some molecular magic. But you make two identical copies. And that is absolutely crucial for what cell division is all about. There's another thing which is very important, is that having replicated these two DNA molecules, and this is, remember that each cell has 23 chromosomes, half of them from your mum, the other half from your dad. So you have some redundancy in there. If mum has a bad gene, hopefully your dad doesn't, and that'll compensate. So 46 molecules of this stuff, and then 2 autosomes, I guess. Or is that included? I can't remember, actually. Is it 46? Anyway, never mind. Numerology is not that important. So these strands separate, but they're held together after they've separated. And that's how you know that one DNA molecule, so you're looking at a single molecule of DNA totally coated with protein to protect it and to fold it up. Because, as I said, you see the cell is only 10 microns long, but some of these molecules are a centimeter or 2 in length. I mean, they're linear. If you stretch them out end to end, they're far bigger than the container. So actually in the cell, they're all sort of wrapped up, and that's what Daniela used to work on. Still a bit controversial exactly how they're wrapped up. Anyway, they're tied together down the middle here by little elastic bands of protein. And that's very important, because otherwise, when you pulled, they would drift apart, and it's very important they don't drift apart until they have to. So let me show you. Look at this cell here. You're looking at the chromosomes now, and you'll see that, choing. Now, I think it's that one that does it next. Nothing appears to happen very much, and then suddenly they shrink, line up, and come apart. And when you see those guys going apart, that's 2 identical sets. So each daughter cell gets exactly the same information. And that's what cell division is all about, how to apportion the DNA so that every cell gets a complete set. And it's done by this thing here. It's done the process is called mitosis. And this is what is known in the trade as the mitotic spindle. Biology is full of terms to learn, and there's no getting away from learning them, because if you don't have the words, you can't speak of them. So the way it works is that there are these things called microtubules here in green, and they're like little ropes, and they're attached at one end to this thing called the pole, rather poorly defined, and at the other end to the chromosomes by means of a sequence on the DNA called the centromere and a protein structure called a kinetochore. And basically, these green things can exert a force on the chromosomes, pulling them apart. And this is a little cartoon of what goes on, for which I paid $15 and from the internet. And I've written twice to the guy to point out his misconception about how it works, because like so many cartoons, it actually has a mistake in it, a pretty serious mistake. His cartoon actually would not work if it was real life, but of course it's a cartoon, so it does work. Things can happen in cartoons that can't happen in real life. The problem is there's nothing holding those chromosomes together. If you see they're being pulled apart, fine, but when they line up, they would already have drifted apart, because he doesn't understand that they need to be held together. So that's fine, okay. But that is sort of mitosis in action for you. And here is Nurse's diagram, which sort of formalizes the thing. The cell cycle consists of a period of DNA replication. 1 chromosome becomes 2 chromosomes, followed by chromosome segregation, in which the chromosomes come apart, and one goes to one daughter cell, and the other goes to the other daughter cell. So that's really what it's all about. But there's a further complexity to this, because this sort of implies that this little cycle here would just go whizzing round and round and round, and you'd have loads of cells in no time. And it doesn't actually work like that. It turns out that for a very long time we've known that if you irradiate cells with X-rays, for example, or shine neutron beams at them or whatever, which damages the DNA, then depending on where you are in this cycle, so this is mitosis when the chromosomes are coming apart. Here, this is where the chromosomes are replicated in what is called S for synthesis phase, M for mitosis. There are gaps in between, and if you shine the X-rays early in the cell cycle, what happens is that you never undergo that switch here. If you shine the X-rays late in the cell cycle, then you never enter mitosis. There are things in there called checkpoints which test the integrity of the DNA, and if things are wrong, the cycle doesn't go around. There are also checks so that if a cell hasn't sufficiently enlarged itself, you'll wait. Basically, it's thought that the control mechanism that decides when to divide is intimately linked to cell growth. And it's in these switches that my interests really lie. I've always been interested in switches where things change. So here's a switch. This is, if you think about it, a bistable switch, a very simple switch. The lights are either off, and if you turn them off, they stay off, or on, and if you turn them on, they stay on. There are other simpler switches where if you press the button, as long as you keep it pressed, the lights stay on. You take your finger off, the lights go off. So you can think of, and of course behind here is just some purely mechanical stuff. Probably two metal contacts are made. It's not very complicated, but you can have a switch doing exactly the same thing, and it looks bloody complicated because this is a radio-controlled light switch. And Christ knows what a 4013 CMOS is. I have no idea how this circuit does exactly the same as what that very simple switch. When I was a little boy, I delighted in making very simple switches where two bits of copper either touched, and they sort of slightly springy, so they tended to come apart. You made a bell push. But you can make very complicated circuits that do exactly the same thing. And then there are also things which are known as latches, and this is a sort of typical latch. I mean, almost every door has one of these things on them. The idea being that it also has two states. I mean, the latch can either be opened, in which case the gate will open, but once you shut the gate, once you slam it shut, it stays shut. And in this case, the horse cannot escape the field, which is important. And if the latch wasn't there, the horse would just be able to come and go freely, which would be bad, if you care about the horse and keeping track of the horse, that is. Maybe it's good for the horse. All right, so how do these switches, these biological switches, work? They have one very curious property. The decision whether to switch is taken with great care. At least that's how we speak of it. That is to say, it's controlled by the things like DNA damage. But once it's been taken, it is absolutely irrevocable. Once you've decided to divide, you go ahead and divide, come what may. Once you start DNA replication, you go on replicating, come what may. You can't start replicating DNA and then decide you've run out of phosphorus or nitrogen, which are two of the important elements involved in DNA, and then say, well, I think this is not a good idea. I need to go back, because you can't unravel the DNA that you've made and start again. So you have to go on, and that can sometimes be disastrous. It's very important these switches decide what to do on the basis. They actually have to measure biological futures. They actually can do that. They can say, well, everything looks okay, and we've got sufficient stores of precursors so that we can go all the way. So in 1961, Dan Mazia, who I showed you already, wrote this enormous long thing about the control of mitosis. And basically the point of this picture here is it's terribly complicated and lots of things are being controlled. But the interesting thing is this sort of starting pistol here, the trigger. And that's the switch, and you can see how much Mazia understood in those days. I mean, it's absolutely, that has no biological meaning. It's just that it looks as though there must be something that performs the switch. And not only, so we don't know how that works, and we also don't know whose finger is on the trigger. That's very important. And actually, interestingly enough, I would say that we still don't know that to this very day. We don't really know what it is that is the final thing that gets a cell to divide. But you can see there are lots of interesting aspects. Now here's a picture of me with fairly long hair, but by a sequence of lucky chances, I ran into this guy here. He was called Tom Humphreys, and he was an expert on sea urchin protein synthesis. And I grabbed at the chance to go and work in Woods Hole during the summers, and these are the people, the students and TAs and things on that course. And they're a pretty distinguished lot. Do you see Ed Southern at the back? That is indeed Jerry Rubin, yep. He's the sort of second in charge of the Howard Hughes Medical Institution. Matt Winkler founded a company called Ambion, which made him a multimillionaire. So we were a pretty distinguished crowd, although I must admit a fair amount of that summer was spent drinking at the Captain Kidd and going dancing at the weekends. But it was fairly work hard, play hard, and it was sort of really, I got to grips with it. So why was I there? Well, I was there to get sea urchin eggs, and here I am getting sea urchin eggs. And it's a pretty crummy sea urchin, actually. I think Arbacia punctulata is right at the northerly end of its range in Woods Hole. And unfortunately, this is a black and white photograph, so you can't see these eggs are a glorious deep purple color, like the purple sea urchin. I mean, you probably, if you've ever been to Greece, you'll have seen rocky shores are covered with either this or another species. So that's a female sea urchin, and she lays eggs. You can't see them because they're not, well, because this photograph wasn't correctly focused, I suppose. But they're about 100 microns in diameter, a tenth of a millimeter in diameter. I mean, large cells by normal standards. And then here's a male sea urchin oozing sperm. And that white goop there is sea urchin sperm. Sea urchin sex is not very exciting, I think, for the sea urchins. And in fact, there is no sexual dimorphism whatsoever. You cannot tell by looking whether a sea urchin is a male or a female. The only thing you can do is give it this little electric shock and see what comes out. If it's purple, it's female. If it's white, it's male. And sex is a matter of this. They're all sitting very closely clustered on the rocks. And as far as I know, it's not known how it is. I mean, it's rather important that they squirt their eggs and their sperm out at the same time, because if they miss each other by a day, they all wash away, and no sperm will meet no egg, and there will be no sea urchins that year. And if you mix, surprise, surprise, the sperm with the eggs, guess what happens? Well, this is why I was there. This is really boring. This is another switch, and it had been found by 10, 20 years before, that when you fertilize the sea urchin eggs, they turn on protein synthesis. We're measuring protein synthesis up the side, time along here, and you can see that the rate goes up about 10 to 20-fold when you fertilize the eggs. And I wanted to know what was going on at this switching point. And it was some time before I actually looked at what was going on down the microscope, and this is what the just fertilized eggs look like. You can tell they're just fertilized because they have this rim around them. That's called the fertilization envelope, and that stops. It's very important that one egg gets fertilized by one sperm for reasons I won't go into them. If the second sperm arrives within 5 seconds of the first sperm, it's disastrous. You get polyspermic eggs, so they're getting the right concentrations. But anyway, this looks like a nicely fertilized batch of eggs. And if I click now to start the movie, you'll see what happens. They divide. Not only do they divide, but they divide amazingly synchronously. And this synchrony is completely natural. We don't have to do anything. Most cells, in order to synchronize them, you have to abuse them in some way. But these sea urchin eggs, you could almost set your watch by it. The first division, this is tremendously speeded up, first division after about an hour, and subsequent divisions every half hour. Even though the eggs are in seawater, it may be sort of 18 degrees Celsius, so much cooler than ours. So they're dividing like hell because, as you can see, a big cell is going down, it's reducing in size. So almost the only thing that they're making new is lots more DNA. So that was interesting, but more interesting, that summer, the summer of 79, this guy, John Gerhardt, came to give a talk. And it was a fascinating talk. He didn't talk about sea urchin eggs, he talked about frog oocytes. Now, what is the difference between an oocyte and an egg? This is important. The oocyte is going to become an egg. And in order for the oocyte to become an egg, so the oocyte is what you would find inside the tummy of a female frog. And in response to the springtime and the presence of male frogs and birds in the air and all that stuff, the females will release their oocytes. They pass down the oviduct and emerge at the other end. As they pass down the oviduct, they encounter the hormone progesterone, which prepares them for fertilization by the sperm. Okay, and I'm now going to add some progesterone to these oocytes. These are about, you can see they're a little bit, just slightly under 1 millimeter. And so these are really enormous, prodigiously large cells. I mean, I suppose the record holder is the ostrich, where, you know, that's one cell, an ostrich egg. That is really giant. So these aren't in that league, but they're still big. They're very big, which allows you to inject them with stuff quite simply. So let's add some progesterone and watch what happens. This is not the most dramatic movie in the world. A white spot forms, and then there's that little convulsion. Now, that little convulsion is actually terribly important because it is the first meiotic division. If your germ cells did not undergo meiosis, every time an egg's fused with a sperm, the resulting zygote would have twice as much DNA as it had before. So basically what eggs and sperm have to do is throw away half their chromosomes so that when 1 and 1 is still 2. So actually in female meiosis, you start out with 4 sets of things and you throw away 3/4 of them and end up with 1. Spermatogenesis is a little bit different, but it's the same sort of principle. Okay, so what is known about the formation of the white spot, and what does the white spot signify? Well, if you look in underneath the white spot, what you find is not a mitotic spindle like the ones I showed you in the cartoon. This is a meiotic spindle, but for our purposes, you can say it's the same thing. And these are fluorescent blue chromosomes lined up on what are no longer green because this picture was taken with different kind of microscopy. And what this Japanese postdoc working in Yale discovered, that what progesterone did was to activate an inactive precursor enzyme that catalyzed cell division. Now, when I heard that seminar for the first time, I was absolutely amazed and bowled over by the idea that you could have an enzyme that catalyzed cell division, because I was used to metabolic enzymes, which did very boring things like put phosphates on glucose or digested your lunch and stuff like that. And this was extraordinary. And the way that Mazia found it was by sucking out some cytoplasm of the white spot egg and injecting it back into a non-white spot oocyte, and then very rapidly this stuff, which he called MPF, a maturation promoting factor, catalyzed the transition, and you actually go through 2 meiotic divisions and end up as a proper egg waiting to be fertilized by the sperm. And there is the first polar body that was produced by that little convulsion. Okay? So what was MPF? What could this enzyme possibly be? This maybe is rather important. Well, the trouble was, and Gerhardt describes in great detail, if you tried to purify MPF, it disappeared. So that wasn't very helpful. And they discovered 1 or 2 things about it, like if you added ATP to the buffer, it helped preserve it, and 1 or 2 things like that. But it was going to, in fact, it was to be many years before the stuff was finally purified, and I'll show you what it ended up being. But along the way, they found out some very interesting things about the properties of MPF. And what was discovered was that every time a cell is in the act of division, in metaphase, where the chromosomes are lined up, and before they come apart, MPF levels were high, and it was easy to detect it. And every time they were synthesizing DNA in interphase between the divisions, you could never detect any MPF. And it wasn't just in meiosis, it was also after fertilization. Every time the eggs divided in mitosis, then again you could detect MPF briefly. And then so it came and it went in a most intriguing way. And it wasn't very long before people working on the same kind of process in starfish discovered that starfish also had MPF, and remarkably, starfish MPF worked in frogs. And it finally emerged that even human cells, if you arrest them in mitosis with certain drugs, they contained MPF, and human MPF works on frogs. I'm not sure that anyone ever tested whether frog MPF would work on humans. So it became more and more pressing to find out what this stuff was, what turned it on, and what turned it off. And I began to think about this and began to think about, what I was really there for. These new proteins here were making new things. And of course you have to make new things because you were an egg and you don't want to just go on being an egg. You want to turn into a baby sea urchin, and that means making new proteins that you didn't have before. That's the whole point. But what had also been found out very early on, if you block that increase, which is easy to do with certain antibiotics, the eggs did not divide. So among the proteins that are made after fertilization, there must be proteins somehow that are involved in catalyzing cell division. And frankly, I didn't really sort of think about that because this was sort of known to the ancients in a way, but it's a good example of how prejudice can blind you to the obvious, because normally when cells divide, of course if you inhibit protein synthesis, it blocks cell division because they need to make more proteins to get bigger. So they need to make more of everything. But in this case, as I said, they don't get bigger, they actually get smaller. So in principle, they shouldn't need to make anything. They've got stockpiles of everything. So that was an important clue. Inhibit protein synthesis. And then by 1982, my studies of how protein synthesis was turned on had been going pretty badly, and I was basically stuck. Completely stuck. One of the problems was that the sea urchins are only ripe for a very brief period, basically July in Woods Hole. And so what would happen, you would think of experiments that you wanted to do during the other 11 months of the year in Cambridge, and then you'd go back to Woods Hole and test your hypothesis. And almost, well, in fact, in every single case, it turned out that the idea was wrong, so nothing happened. So it wasn't a very good way to do biological research, actually. It's better to have your material there all the time. But there are no sea urchins in Cambridge, and if there had been, they wouldn't be ripe, and so on. So I started reading books, and I read this book by Jacques Loeb about artificial parthenogenesis and fertilization. And having had a good religious upbringing, I was sort of intrigued by this. Here is the Holy Spirit descending on Mary's womb from Fra Angelico. And I thought, what a good opportunity to do a kind of molecular religious experiment. And I wanted to compare the patterns of proteins made by parthenogenetically activated sea urchin eggs with properly fertilized sea urchin eggs, because there was some reason to believe they might be different, which might shed some light on the possibility of virgin birth. Anyway, and there are poor old Adam and Eve being expelled from the garden. Anyway, so I did the experiment. I'm going to show you one half of the experiment. Now, this is where the ambassador definitely loses it, because this is a one-dimensional SDS polyacrylamide gel. And this is how, for many, many years, we've been analyzing proteins. It wasn't available when I was starting out as a graduate student. It's a very simple method, which I won't describe. But basically, all you need to know is these are radioactively labeled proteins made visible by a process known as autoradiography. And each of these bands here, there is a protein, there is a smaller protein, here is a very small protein, here is a great big protein that's sort of stuck at the top of the gel. And you can see each one of these little visible bands is a protein. So easy to compare patterns of protein synthesis.
But what I noticed was that this protein here is easily the strongest one made at early times. It's 26 minutes after fertilization, probably 20 minutes after adding the label or something like that. It gets stronger and stronger, and then it faded away, and then it actually comes back again. Let me show you. This picture here is a little bit easier to see, I think. This is a different organ. This is clams rather than sea urchin, but the principle is exactly the same. You can see these proteins coming and going, and they kept on coming and going every time the cells divided. So now at the time, this was impossible. Proteins did not come and go like that. Proteins in your stomach, sure, they go because they get bathed in digestive enzymes, but you can't have. So I looked at that, and the idea that this might have something to do, I didn't think it was actually MPF itself, but I thought it might be something to do with the control of turning MPF on and off. It's obvious. So here's when you would find MPF in these cells, and here's the behavior of the protein that I called cyclin. And there were two things that made me think that it couldn't possibly be MPF, actually. One was that we tried to calculate how much of this stuff you made, because you've only got half an hour, in this case, to make it. And you can't make very much protein in half an hour in a fertilized sea urchin or clam egg. But I calculated roughly, so using some back-of-the-envelope considerations, and actually, it's of the same order of magnitude as an enzyme called glucose 6-phosphate dehydrogenase in a red blood cell. So I mean, it's not negligible, but it's not huge. And the other problem is it goes up steadily, whereas MPF appears suddenly, and that also bothered me. And actually, that's still something that we don't understand very well, as you'll see. So I thought this was way more interesting than what I had been studying, and I started to ask people, if they could see anything wrong with the idea that you made this protein and you turned on MPF, and you got rid of it and you turned off MPF. And nobody could see anything wrong with the idea. And I got very excited about it. I knew in my bones that I'd made an incredibly important discovery, which was totally, what was interesting about it in a way was that although, this, if you're used to looking at these patterns, I mean, this is absolutely compelling. There's no escape from it, really. But if you read the books, or if you read the books on the control of the cell cycle, nobody had ever in their wildest dreams had imagined that something had to go away in order for cells to divide. And that's what I seem to be saying. Unfortunately, we had no way of stopping it going away because we didn't know what the disappearing mechanism was. And actually, it's still pretty hard to do. So I began to read about the cell cycle, and this is Lee Hartwell, who worked on yeast and had discovered that there were genes that controlled cell cycle transition. So here's a gene called CDC28 that controls a process called START, which turns on DNA synthesis and the formation of this little bud here and something inside called the spindle pole body. And here's a gene at the end which allows the separation of the two cells. And if you mutate these genes, then you find, in this case, no buds on any of the cells and only the final amount of DNA. Here, on the other hand, you find that all the cells end up looking like this. And if you just inhibit, on the other hand, protein synthesis or DNA synthesis, you see something more like this, which is that some of the cells have big buds and some of them have small buds and some of them have no buds. And it's much more heterogeneous. So Lee was really the pioneer here, but he didn't know what any of these genes did because this was before DNA sequencing. So you really couldn't tell, and you couldn't possibly deduce how they acted. So instead, he did very beautiful formal genetic analysis to show when the genes acted, and that was his great strength and interest. And then Paul Nurse read Hartwell's papers and was inspired by them, decided to work on a different yeast. It's called fission yeast. And again, at the non-permissive temperature, a cell division cycle mutant can't divide, so the cells just get longer. And here's a CDC2 mutant. And Paul happened to notice one day, he was trying to enrich for these kind of mutations by centrifugation, he saw these small cells, and he realized, looking at them, that what had happened here was that when you knock this gene out, the cell cycle actually advanced, and the cells divided prematurely. And he thought this was really interesting because they must represent a sort of controlling mutation. So he went looking for other genes that could be mutated to give this same Wee phenotype. And the amazing result was that the other thing that he found was a gene called CDC2. So if you kill CDC2, you never enter mitosis. But you can also hyperactivate CDC2 so that you accelerate this transition. And then in Lee's case, the gene called CDC28 clearly catalyzed somehow, was responsible for switching on DNA replication. So it came as a little bit of a surprise when these genes were finally sequenced that they were in fact interchangeable and basically the same thing, and they controlled both transitions, both entry into S phase and entry into M phase. And you could either kill them or hyperactivate them. And the sequence immediately said that they encoded a protein kinase, which is an enzyme that sticks phosphates on other proteins. But it had no activity. And then it was found in humans, and that created great excitement because nobody realized that actually yeast cell division and human cell division were so intimately related. And nowhere does cyclin appear in any of this. In fact, cyclin was not spotted in yeast at all. So here's a typical example of a protein kinase. Actually, this is the CDC2 protein kinase, but sticking this phosphate on this little residue here makes this thing 100 times more active than that thing. And you've got jolly good eyes if you can see any difference between these two structures. So they're very subtle, these alterations, but incredibly important. I mean, 100-fold is an awful, it's basically dead and active thanks to that phosphate. So but phosphates don't always do that. Sometimes they can equally well turn things off. Sometimes they stick things together. Sometimes they blow them apart. It's rather hard to predict. Anyway, it turned out that from Paul's analysis that the way Wee1 worked, it was also a protein kinase, and it stuck an inhibitory phosphate onto CDC2. And then there was an enzyme called CDC25 that took that inhibitory phosphate off. So the control of CDC2 was getting pretty subtle. And it ended up, when MPF was finally purified from frogs, it turned out to be a dimeric enzyme. It had two subunits, one of which was CDC2, and the other one was CDC13, and CDC13 from its sequence was clearly a cyclin B. So, and then you can just do the very simple experiment, just mixing these two things together, and you could show that indeed CDC2 was completely dead until cyclin bound to it, and then there were these other controls. Now, this is where the complexity comes in. I mean, already it's beginning to look a little bit complicated, but it's even more problem. I like to call this the Parmenides problem. Because you remember Parmenides said, what is, and what is not. And it's kind of hard to argue with those propositions. And he therefore concluded that it was impossible for what is to become what is not, and what is not to become what is. So everything you see, I mean, it looks like I'm waving my arms, but you're deluded. And Parmenides said that, or at least as far as we can tell, because his traces are rather fragmentary, that the world was a homogeneous, changeless sphere, and nothing ever changed, really. So this is a good example of a bistable switch, because it turns out there's a reflexivity. Wee1, I've told you, inhibits CDK1. That's the other name for CDC2. CDK really means cyclin-dependent kinase, so that's that's CDC2 plus cyclin there, okay? But when you enter mitosis, then it turns out that Wee1 is turned off. So there are only two stable states for this situation. Either the kinase is active, this kinase is active, or that kinase is active. And if this one is active, you're in interphase, and if this one is active, you're in mitosis. But how do you switch between the two? It appears to be impossible. So that is a problem which is still very much with us. And when you look at some of the other kind of control processes, I mean, I don't try and remember this because I don't even understand it, and I don't think anybody does really. I mentioned that damage to DNA caused an inhibition of entry into mitosis. This is a partial diagram of the circuitry that controls that, and it's very, very unclear how it actually works, and there are a lot of other protein kinases, and there's a lot of reflexivity. So let's forget about these details and just say that basically it's incredibly simple. The cell makes cyclin, MPF turns on because the cyclin joins to CDC2, the kinase is activated, and the cell enters mitosis. The nucleus breaks down, the chromosomes all line up, and everything is hunky-dory. Then the cell magically degrades cyclin when all the chromosomes are lined up, MPF turns off, and the cell exits mitosis. And here the chromosomes are all lined up, and now the cyclin is gone and they come apart. And in fact, the cyclin goes about 90 seconds before you see the chromosomes come apart. And if you stop it going away, then the chromosomes don't come apart. Now, so far, so good. But, and it's a big but, as far as we know, these enzymes can only do things by phosphorylating their target proteins. So if you're a biochemist, you want to know how many substrates have to be phosphorylated, how much, in order for a cell to enter mitosis. I mean, it's a simple question, very complicated answer, because it turns out that there are hundreds of substrates, and many of them have multiple phosphorylation sites. So you have to ask the question, in order to alter the properties of this particular enzyme, is it important that all 4 phosphates are on there, or will 3 do? Is the combination of sites, is it sort of 1, 2, and 3, or 3 and 4? And no, I mean, it's difficult to find that out. But it's catalyzed by CDC2 and cyclin. Okay, so that's fine. We can accept that, and we can accept that it's going to take a while to identify all these protein substrates, and even longer what it actually does to those proteins, because that's what we would really like to know. But then there's a new problem has arisen here. How do you get out of mitosis? Because if it's true we went into mitosis by phosphorylating all those proteins, somebody has to take all those phosphates off. And this now presents a big problem, especially since geneticists and biochemists disagree on the point. So this is an enzyme called protein phosphatase 1. The phosphatases are enzymes that remove phosphates. And this is a quite different enzyme called protein phosphatase 2A. And it's a little bit odd that there is so much disagreement between perfectly good evidence on both sides. So usually the answer to that is they're probably both right, actually. But because of the way they look at it, they get a different answer. But of course it raises, if you think about it, this is an absolutely sort of classic biochemical problem. If you have an enzyme that's sticking a phosphate on at the same time as a phosphatase taking the phosphate off, then it's rather hard to see how you can get anything fully phosphorylated, because as soon as you pop this on, off it comes again, and all that really happens is that you hydrolyze a lot of ATP and the cell gets hotter. So it really seemed to me, from first principles, that there ought to be some way when you turned on the kinase to turn off the phosphatase that got you out of mitosis, and then somehow at the end of mitosis, you would reverse the thing. And interestingly, I sort of didn't believe it strongly enough to suggest that anybody actually work on it, and we were eventually led there. So the principle is terribly simple. If you want to put water in your wash basin, you must turn on the tap. No tap, no water. But you also have to put the plug in. I mean, this is, you see, we can be very simple. This is not complex, actually. This is simple. And the tap is the kinase. You see there's a slight asymmetry here. The phosphatase is the plug, okay? So that's how I can sort of remember these things. So Satoru is the hero of all the experiments that are going to come. And he exploited a system which had actually been used in the purification of MPF by Fred Locher and Jim Mahler in Denver. And what Fred had discovered was you take a tube full of frog eggs, there they are, packed tightly, and you simply spin them, not terribly hard, but hard enough to crush them. And you get this picture here where this yellow layer here is absolutely pure, undiluted cytoplasm. And it behaves exactly like the cytoplasm would behave in the cell. It doesn't know that you've taken it out of the cell. It sounds a little bit brutal, but actually it works beautifully. And if you add nuclei, some DNA-containing nuclei to this extract, that's what happens. I mean, the extract will assemble these gorgeous meiotic spindles. There are the chromosomes, there are the two poles, and here are all the microtubules joined together. This is, I forget who made this lovely picture, not me. And actually, if you add calcium to this, which replicates the arrival of the sperm, every time a sperm hits an egg, there's a big increase, transient increase in calcium, then the chromosomes would come apart. So it'll do everything. It'll do cell-free cell cycles. It's marvelous. So we started looking at, for reasons I won't go into, we started worrying about this phosphatase problem, basically because we saw a phosphatase come on. And we decided to measure kinases and phosphatases, or rather to look at this question of whether the phosphatases were regulated. And what you can see from this experiment here is that if you measure the kinase, it really goes up quite gradually, rather like this. The books suggested that what happened, it didn't go up, and then this WEE1 CDC25 switch suddenly switched. So you got a sudden huge peak of kinase activity that simply overwhelmed the phosphatase, or that was the implication. But if you look at the data, it doesn't look like that. You can see cyclin accumulating. You can see here a protein undergoing a little hop, skip, and a jump on the gel because that tells you that it's getting phosphorylated, that little gel shift. Okay, so it looked more like the kinase accumulates gradually, and there must be a high phosphatase activity which suddenly switches off and then comes back on. So we decided to assay phosphatases. So Satoru designed a substrate for phosphatases. And again, I won't go into the technical details, but basically you make a protein with a phosphate that resembles a mitotic phosphate, so you can add it into extracts. And when we did that, we found the most amazing thing, which is in an M phase extract, a mitotic extract, there is no activity against that substrate. And when you activate the extract by adding calcium, you get a tremendous peak of activity. It goes down again, then it comes back up again. And actually, it's this second appearance of activity that really does the business of getting you back to interphase by taking all those phosphates off. And we can show very easily that there are actually two enzymes here, not one, because certain inhibitors, this is the stuff that gives you shellfish poisoning. And the first phosphatase is not inhibited by it, but the second phosphatase is. And the other way around, this is an inhibitor with exactly the reverse specificity called cyclosporin A. It's an immunosuppressive drug. That wipes out the first peak, but the second peak now appears. So we needed to find out what this second peak was, and were the geneticists right or the biochemists right. But before we did that, we had a look to see whether indeed it was correct that this phosphatase really did turn off in mitosis. So this is an extract coming into mitosis, and you can see that here's the phosphatase activity, and it suddenly switches off when the cell extract enters mitosis, comes back on again. So this was fantastic because it shows that they're really, as well as the kinase going up and down, the phosphatase is behaving in a totally reciprocal manner. So that's incredibly good. So we next needed to find out what this phosphatase was and how it was turned on and off. And this story has a happy ending. After several years of looking, we discovered that the phosphatase was this creature here. It's called PP2A B55 delta, not a very romantic or helpful name, actually. But basically, this is the enzyme bit. This is a sort of scaffolding that puts these two bits together, and probably this bit of the protein decides what kind of phosphorylated substrates this enzyme likes to deal with. And how was it regulated? Well, it might be that you took this B subunit and put a phosphate on it, or it might be that you stuck what is known in the trade as a pseudo-substrate a protein with a phosphate on it, which didn't quite fit right, so that the active site of this thing couldn't quite reach it. So it inhibits because it occupies the site of selection on the B subunit, but it can't be degraded. This is known in the trade as a phosphorylatable phosphatase inhibitor because it's only inhibitory when you stick a phosphate on it. And it turned out that was the right answer. And the way we got there was rather roundabout because it turned out there was another enzyme, another of these protein kinase. You begin to get the feeling there are an awful lot of protein kinase. Phosphates are being put on and taken off lots of proteins in order to regulate this. And indeed, this kinase its sole function, as far as we know, is to act as a phosphatase inhibitor. Oh my God, the reflexivity is getting a little bit complicated, embarrassingly so almost. And Satoru was able to identify the substrate of this great wall kinase, and it was a small heat-stable protein that stuck a phosphate on here. And these two things, they're two very closely related small heat-stable proteins, were potent inhibitors of the phosphatase whose structure I showed you. And then when we found that, we looked at the literature on these things, and this was a wonderful case. Normally when you find a new protein, you find a vast literature on it, and you don't know where to begin. In this case, there were only something like 20 papers in 15 years, so even I could read them. And one of them said that we were clearly right because one of these little inhibitors called alpha-endosulfine, a conserved protein required for oocyte meiotic maturation in Drosophila. Fantastic, that's exactly what we want. And this woman, Daniela Drummond-Barbosa, had been interested in this because of a misconceived idea that this had something to do with the control of insulin metabolism, which actually it doesn't. But they'd done some very nice experiments, which, because they were geneticists, they couldn't interpret correctly. Very interesting, that. I mean, I'm not very good at interpreting genetics accurately, but the reciprocal is also true. And what they showed was they had high CDK1 activity, yet the oocytes couldn't maintain themselves in meiosis, exactly as though in this strain of flies, you turned on the kinase, but because the phosphatase was not turned off, you were just having a battle, and actually it was the phosphatase that won, and that's why they couldn't do it. So we then asked, is this for real? And other questions like which comes first. And basically, here is the endosulfine getting phosphorylated as the extract enters meiosis, and we were able from this to measure how much there was of it, and there was enough to inhibit all the phosphatase, so that was very satisfactory, and it all gets phosphorylated when the extracts enter mitosis. So everything checks out, so there's absolutely no doubt about it. And if you take it away, which we can do, then here's a situation where the kinase turns on, but the phosphatase has not been turned off. And you can see, normally it ought to look like that, or these bands ought to be that black. And I think you can see, maybe it's easy to see, compare that blackness with that blackness. You can see this is an awful lot blacker. And that is an awful lot less than that, right? Here we've gone 90% up. Here we've gone 10, 20% up at most. And that's because the kinase is sticking on the phosphate, but the phosphatase is taking it off again because we've removed the inhibitor of the phosphatase. It's very difficult, I find, to remember when you start talking about inhibitors of inhibitors. It's really easy to get confused. So let's skip over that. So this is where the problems began. We think we've defined the circuitry. And what I do at this point is go and consult my friend Bela, who is Hungarian in Oxford, who knows about the control of mitosis. And here you can see him fretting about WE1, and here's B55, and all kinds of things are zooming up and down. And what Bela pointed out was the circuitry, if you draw it out, looks like this. And this has some interesting features. So here's the CDK WE1 positive feedback loop. I mean, so this is very difficult to model because you don't really know, as I pointed out, which way it's going to flip. And it turned out later that the control circuitry between endosulfine, the inhibitor of this enzyme, that's what that means, this is also an inhibitor. In other words, when these get phosphorylated, it's this enzyme, the very one that inhibits, that reverses it. And what is even worse is that we now have the CDK being regulated by these phosphatases and kinases, and we have the downstream targets being phosphorylated or dephosphorylated by this enzyme. And this very simple-looking circuit actually behaves in a very difficult-to-predict kind of way. And life gets even more complicated. At this point, basically, I retired and stopped working on the problem, but people went on working on it. And there are some new facts, and the new facts are extremely difficult to understand. So the first good fact is that Great Wall, the thing that phosphorylates the endosulfine, is an essential gene. That's good because it means it's important. If you don't have it, the mouse is dead. But you can make, by genetic trickery, this is a guy called Marcos Malumbres who's very good at this kind of thing, you can make somatic cells from these baby mice and then take the gene out. And the surprising and, to me, absolutely shocking result was that these cells have absolutely no problems entering mitosis, whereas the frog eggs did. So that's very peculiar. And then even closer to home, mouse oocytes can be made lacking this system. They lack Great Wall. And they, in response to the hormones that control mice as opposed to frogs, they enter meiosis absolutely normally, I mean, with no difference whatsoever. And they complete meiosis 1, but after that, they crash out of meiosis. So there seems to be a difference between somatic cells and their dependence on this system and oocytes, which is really quite mysterious. I could kind of justify it. And then, to make matters even more confusing, it turns out that yeast have exactly the same system, but it doesn't control mitosis or meiosis at all. It controls survival in the face of glucose starvation rather than, and it's absolutely identical system. So it looks like what nature has evolved here is a plug-in module that you can use to control certain things, but what you actually use it for changes depending on the kind of cell or the kind of organism you are. And frankly, this, at the moment, doesn't make sense. I mean, there's something we're missing. Baylor actually rang up. He has a habit of ringing up at most odd hours and odd places. And I was driving to work last week, and he said that a friend of his in Oxford has just done some wonderful experiments, which I will like very much, but he didn't tell me what they were. So, but basically, what I've said, this is the way it works. Proteins without a phosphate get phosphorylated, and that's how you enter mitosis. It's catalyzed by this thing here, CDC2 plus cyclin B. In addition to that, however, CDKs, it turns out, phosphorylate Great Wall, which phosphorylates ENSA, which turns off the phosphatase, which then means it's much easier to put all these phosphates on. So again, and when the cyclin gets degraded and this arrow goes away because the CDK turns off, then quite rapidly Great Wall gets dephosphorylated, but it doesn't happen immediately. So one idea is that it's actually a clever delay timer, and the whole idea is that you sort of stay in M phase for a little bit after the chromosomes come apart. And the idea is that you don't quite get out of mitosis until you've had sufficient time for the two sets of chromosomes to get that far apart, and then you can divide the cell in two, and maybe that's part of what it's all there for. But the answer is we really don't know. But in biology, if you don't know something, you just, as I tell the graduate students, just do the fucking experiment. Because they tend to think that you can deduce it all from first principles, and actually you can't. It's really, really important to do the experiment. Now, back to Gilles de Gennes. So this is the communication difficulty. That's all I'm going to say about the science. Science is clearly a form of art with the same invention and the same doubts. There are major differences, however. One is the difficulty of communication. An Indian playing his flute in the streets of Bogota invents a new tune. Within 10 seconds, a passerby may be struck by it, possibly for their whole life. But in our trade, A beautiful discovery can be transmitted only to people who have been through a long, specialized education. We must do our best to keep in contact with our fellow citizens, but we often fail. A lot of this stuff, I mean, since I don't understand what's going on, how can I possibly expect you to understand what's going on? I can only tell you what I know. And I sort of like this, you know, the difficulty of extracting a single vision from a scene, making an impression. Simulations and numerical exercises, the analogues of photography. What we need is a simple impressionistic vision of complex phenomena, ignoring many details. This is very good, I think, you know. There are a lot of people who think nowadays that if you just accumulate all the data you possibly can about some process, that the truth, the understanding will sort of come and punch you on the nose. And I don't think that's true at all. You have to have ideas about what's going on. And finally, he says, incidentally, the artistic profession suffers from many parasites, among others, the art critics or commentators. Fortunately, we do not have the counterpart of the art critic in our sciences, although some referees tend to mimic this style. So here is an emerging pattern. Actually, this isn't much of a pattern. This really is a pattern. If you've ever approached Heathrow from the Windsor Castle side, you will have seen this avenue of trees. I mean, that really is a pattern. It's fully emerged. But as I flew here yesterday, I saw these clouds, and I wonder, you know, toing, 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 toing. So what's all that about? I have no idea. Thanks for listening. End of show, it says here. So if anybody's got any questions, I will happily entertain them. But usually I stun the audience into complete silence.
There's one. Weird. What's a somatic cell and what's an oocyte?
Well, okay, good question. A somatic cell means a cell in your body, whereas an oocyte is an egg. I mean, a cell is going to be an egg. So we distinguish between germ cells and somatic cells. So again, that's a sort of concept, that's a 19th century concept. And it actually is a sort of quite an important and deep concept if you think about it, because I said right at the beginning that, you know, we're all descended from some cell. And there's no getting around that. I mean, that really is an article of scientific faith. But what's funny about that is that you know and I know that we are mortal. We're not going to last for 4.8 billion years or whatever it was that we heard this morning. So what is continuous? And the answer is it's the germ cells which are continuous. So how do the germ cells avoid the damage that, alas, eventually attacks my knees and my heart? And I think the answer is probably selection, actually. I mean, you know, when a fertilized egg develops, if it develops into an adult, that means that everything was okay. And if it doesn't, well, those genes, those chromosomes will be lost to the population. What is slightly unclear is whether there's any, you know, you might say, well, there must be a checking mechanism that operates during the formation of the germ cells. You could imagine a little man going along and checking the DNA to make sure it's okay, but I don't think there's any evidence that that actually happens.
It just makes me think, which came first, the chicken or the egg?
Oh, absolutely. No, indeed. And also, actually, the quote I like about Lewis Thomas, who was a physician who used to write a column, I think, in the Boston Post or something like that, and wrote some very lovely essays, said at one point, you know, the important thing about DNA replication, it should make some mistakes, because if it didn't make any mistakes, there would be no evolution. And if it didn't make any mistakes, there would be no music, and we would be anaerobic bacteria.
Yeah, I would like.
Sorry.
So I would like to ask a question about the mechanism by which you do this research in these gels. You know, the gels are they are expressing a certain protein, and you
well, the sample has all these proteins in it.
But you know the lines are labeled. Do you know to which protein they correspond?
Sometimes and sometimes not. I mean, some of the ones, you know, we focus on the ones we're interested in. But
you can select them.
You can select them. I mean, typically what we use is an antibody that's been directed against, so it only recognizes that.
Okay. So these lines are labeled, and you know how they evolve during a certain process.
Or you can sometimes cut them out of the gel and give them to your analytical chemist friend, and he will tell you, or she will tell you what they are, and then you can sort of work backwards from there. But it's quite tricky. I mean, it's not straightforward. But an awful lot of what I do, anyways, depends on that.
And the research proceeds by knocking some of them off and then repeating the same experiment, or
it varies a lot, actually. I mean, there are. I mean, in my case, mostly we knew what these things were, and we were worried about their behavior. But other people, I mean, you know, the best thing in the world is to knock them out and make sure they go away, because sometimes that doesn't happen, and you're mistaken about
your particular discovery was this cyclin?
What I discovered was that cyclin went away. I mean, it's really hard to explain that to your grandmother, right? I won a Nobel Prize for finding something that vanished. Yeah. You know, it's sort of weird, but that was really what it was, because at the time, it would have been it was inconceivable. Nobody would have said that you could so specifically. I don't know if I didn't really point it out, but you know, this protein goes away. All the other proteins are absolutely fine and unaffected. So that was considered to be impossible at the time. No known mechanism could possibly lead to that, because when you added the kind of enzyme that made other proteins go away, everybody would get destroyed. So that you can't.
Thank you very much.
But there he was in black and white. I mean, as you saw, there was no other possible conclusion.
Thank you very much. It was very, very exciting.
So I'm going to ask a different kind of question. Obviously, the underlying lab work and all that is very messy. But what's striking is the quality and clarity of the graphics in your slides. And they are deeply aesthetic and fantastic. And so my question is that part of the research project in your mind?
Well, it's part of giving talks and trying to clarify it in your own mind. I mean, some of the, I probably can't get it back. You have an interesting point there, actually. Let me go back to one slide. Come on, where are you? I know it's in there. Oh yeah, here we go. I think this is a good example of a bad slide, in my view, because what it serves is to try to clarify the mind of the slide maker rather than explain how things work to the audience. And because, you know, you look at that and you say, Christ, what's going on here? And, you know, I mean, it's not the world's worst slide. We could get you some wonderful examples of truly bad slides, but it's really not very good. It's too complicated. And I do like to try and make, I think I would have loved, if only I could draw, I would have loved to have been an artist. But I can't, so I'm not. But I do think that, you know, trying to, and I get a lot of, I mean, actually one of the good things about being a Nobel laureate is you get asked to explain things a lot. So you can actually try out different things and, right? And refine things and realize that there are, I mean, it's amazing. Actually, I mostly learned that I should really put a plug for the problems book, which is outside. So we did the illustrations for that. I learned an awful lot from my friends who are professional illustrators about consistency and clarity and trying to pare away as much of the irrelevant details as possible.
Well, even things like the choice of the colors of the slides.
Oh, that's sort of important too. Yeah. I mean, my friend Keith Roberts, I forget, his uncle knew Jim Watson. So Keith went to work with Jim in his second year as an undergraduate. And Jim discovered that Keith wasn't so great at lab work, but he was absolutely brilliant scientific illustrator. So he got to illustrate Jim's first textbook. And, he got really, really good at it. So normally when we're writing these books, we're all in the same room together, and I look over Keith's shoulders and learn a lot from that. And then there's another one called Nigel, who is very good at the computer graphics side of things. So, you try and, it's good. Seriously good. Fun.
I have two questions. The first is related to origin of our mankind, to evolution of human. I remember for some time there were theories that the fact that in Africa there is, in some territories, there's increased level of radioactivity. It could increase rate of mutation, and in this way, evolution, let us say, of human could be influenced. What is now the stage of art on this concept? Is it valid?
I've not heard that hypothesis. What is for sure is that when you sequence Africans, there is far more diversity there than there is in our populations. And I think the interpretation of that is that when humans first invaded North Africa and the Arabian Peninsula and then on into Europe, the populations were very small, so it had a sort of effect of sort of homogenizing everybody thereafter. But I don't know, I doubt whether that I mean, it's true, radioactivity is one mutagen, but there are some much worse things out there. I mean, I learned from my former boss, Thomas Lindahl, that actually the two worst mutagens, the two biggest enemies of the integrity of DNA are oxygen and water. Okay, and they are responsible for something like 25,000 mutations every day in every one of your cells. And that's, I think, probably why we age. If there was no oxygen and no water, we'd last a lot longer. Well, we wouldn't actually, of course,
And would you allow me the second question?
Yeah, sure.
About the control of expression. We know that we have pairs of genes that can represent a feature of our organism. And there are another genes that control which of these genes are expressed. So what controls
the controllers?
What controls the controllers? Is it
several levels? It's a hierarchy of things. I mean,
is it several levels of such?
Yeah. So I mean, it's easiest to answer that question from flies, because this is where it was really first discovered. So when a fly egg is formed, it turns out to have a gradient of a particular protein, high at the front and low at the back. And that sets up the first differences in gene expression between the front of the fly and the back of the fly. And then this pattern is successively refined by smaller and smaller scale gradients of regulatory proteins. I mean, there are some people who really give a pretty complete, I'm not sure how accurate it is, but a guy called Eric Davidson, for example, has looked at the early development of sea urchin eggs and sort of knows what some of the circuitry is. So you get genes, I mean, it's very complicated. You get genes that turn one gene on and another gene off. So just looking at a protein, you can't tell whether it's having an effect one way or the other, but you can do experiments to deduce this. But I don't, this is sort of gets into finer and finer detail, and I probably, in the case of human beings, not so much is known. I mean, most of the information in humans comes from studying cancer cells, which make too much of something or too little of something. But that's not very interesting in terms of deciding what makes a difference between an eye and a toe and a liver and a lung and all that kind of stuff. And we don't even really know how many different kinds of tissues there are. Most people would say about 250 different kinds of cells, but that's a number slightly plucked out of the air. But it's a jolly interesting and important question. That's what it's all about, getting information out of DNA.
So one of the most striking things about this whole process is not that there are mutations, but how infrequently actually mistakes are made.
Well, it's pretty amazing how robust the system is. Yeah.
So do you, or is it known at what stage the primary error checking is done?
Well, I think one of the ways the error checking is done is that if you detect a really serious error, you die. You can, I mean, not you, but the cell. Because cells are very expendable. I mean, and there are marvelous mechanisms that if something goes horribly wrong with a cell, perhaps because of mutation, then it simply commits suicide and its neighbors will eat it up and recycle all the components. And that's absolutely fine. In other words, doing things right is more important than having enough resources. A lot of people think, oh gosh, that would be awfully wasteful. And it turns out that that's a very weak argument in biology. And whenever you look at something and it looks like it's incredibly wasteful, it's probably more important is sort of getting things right is the important thing, not waste. But so that's a good question. I mean, there are some things which are amazing, like the tendons in they're incredibly old. They don't really turn over, and that's one of the reasons, I guess, why your knees. I'm particularly keen on knees because mine are not very good. It's very difficult, apparently. Basically, you've tied these ropes to your knees, and it's then very difficult to repair them. So some things can be repaired quite well. I mean, your skin is falling off all the time to be replaced by good cells, and if the cells can't reproduce, they'll just get out-competed. I mean, that's what happens. Phil actually is the person who really should be answering all these questions. Not me. Okay. What do I know?
Okay, thank you. I think we close there, and it's my pleasure to give you a little memento of this.
Oh, wow.
Let's thank
I get there.